So I'm going to be talking about bacterial antibiotic resistance on Earth. And since human beings have polluted the Earth a lot, we are now, thinking about colonizing space. And as you know, there have been a number of space expeditions. And under microgravity, human physiology changes. Immune response is compromised. And so the concern about antibiotic resistance of bacteria is even more serious in space than it is on Earth, although it already is a very serious problem on Earth. So this is a highly collaborative effort. Lots of people are involved, as you can see, and I'm not going to name them. That would, exhaust all the time I have. But the slide is here, and you can check it whenever you want. And the papers with which these people are connected are also listed on this slide. So this gives you an idea of the time frame in which different antibiotics were deployed, and the resistance. The top part is the time in which the antibiotics were deployed, and the lower part is when the resistance developed. And from 1930 to 2005, if you go over it, you see that the interval between the deployment and the development of resistance became less and less. And the reason for that is overuse of antibiotics and using it for frivolous purposes, such as fattening the cattle and things of that kind. So as a result, Now, the antibiotic resistance is developing. Many of them are becoming ineffective. And yet the introduction of new drugs has been declining. And from 83 to 87, something like 16 new drugs were approved. In 2008 to 2009, just one. Okay, so question is why and how this resistance has developed. So for that, I have to briefly discuss the established mode of action of bactericidal antibiotics. So beta-lactam, for example, penicillin, interfere with cell wall biosynthesis. Quinolones, for example, ciprofloxacin, interfere with DNA synthesis. Aminoglycosides, such as gentamicin, interfere with protein synthesis. And now what bacteria have done is to develop resistance to these various interferences. There are various mechanisms by which antibiotics act upon them. And a potential solution then is to find alternate targets for making bacteria sensitive to antibiotics again. And we have done just that. And that mechanism is to weaken the antioxidant defense of bacteria. And as you know, the antioxidant defense protects bacteria against reactive oxygen species. The examples are superoxide and hydrogen peroxide. So to introduce the subject further, I have to again give you a little bit of background with which I suspect most of you are familiar. As you know, the transcription of the genes that are present in DNA of bacteria or anybody else takes place through RNA polymerase. And here is you can see my mouse, I suppose. This is the RNA polymerase core enzyme, which by itself can do very little until it is combined with a small protein called the sigma factor. And once the two are combined, the two are together, you call that a holoenzyme. And what particular genes the RNA polymerase transcribes depends upon which particular sigma factor it is combined with. And what we are concerned with here, I won't go in, there are several different sigma factors, and I won't go into all the list of this. We are concerned here with sigma S, which is encoded by the RPOS gene. And sigma S RNA polymerase transcribes genes that make bacteria resistant to many stresses. And the examples are given here: ethanol, hyperosmosis, heat, oxidants, starvation, and such. Now, how do the proteins that result from the action of a sigma S RNA polymerase, after the transcription by this polymerase, how do they protect? Some examples are given in here, the examples of the proteins involved and what they do. There are molecular chaperones. Examples are DNA K protein family that protect proteins. This particular endonuclease protects DNA, trehalose enzyme membranes, alanines, carboxylase, cell wall, and so on. You can see the antioxidant proteins protect against oxidative stress. So as you can see, these are the very, the biomolecules that these proteins, these stress proteins protect, are precisely the same molecules, as you may have gathered from the previous slide, that the antibiotics damage. So we reason that just as sigma S encoded proteins protect bacteria against these various stresses, sigma S RNA polymerase, sigma S itself, should be involved also in protecting against antibiotics. And that turned out to be the case. And here I have given you examples of 3 antibiotics. And the bacterium involved is a clinical isolate. We got it from the Stanford Hospital. Clinical isolate of uropathogenic Escherichia coli, UPEG for short, which causes cystitis and other urinary tract infections. And you can see that, these are all, the top lines are all controls, which you don't need to pay attention to. The point is that in each case, if you eliminate the gene that encodes sigma S from the strain, that strain becomes highly sensitive to killing by any of these, all of these antibiotics, individual antibiotics. So indeed, sigma S is involved in protection against these bactericidal antibiotics, which are the center of the focal point for treating bacterial infections. So we wanted to know what is the mechanism by which sigma S is involved in protecting, which particular proteins are involved. So for that, we did a 2D gel analysis of the wild type containing sigma S and the mutant missing sigma S when they both were exposed to gentamicin, one of these antibiotics. And we found, of course, several different proteins that were affected, but in particular, proteins that are involved in antioxidant defense. And I've given you an example, 2 examples, superoxide dismutase and transaldehyde, something like over 30 or something 33-fold less, level in the sigma S mutant exposed to gentamicin compared to the wild type. So the wild type is enhancing its capability to deal with the oxidant defense, which the mutant is unable to. In fact, its capacity to do this is minimized. So the results suggested that as I just mentioned, the GM treatment caused greater oxidative damage by ROS generation in the mutant, missing sigma S. And what I'll do for the sake of economy of time is to go over very quickly the slides that would show this fact that ROS are involved. And this will be done by showing the induction of a gene that is induced by ROS, dyes that measure ROS, for example, MPLEX red, damage to proteins expressed to reactive oxygen species, carbonylation that is, protection against gentamicin by antioxidants, increased GM sensitivity of mutants, missing individual antioxidant protein, not just sigma S, but the individual antioxidant protein that sigma S controls. And then to show that, and also this NADPH I told which is needed by the antioxidant proteins to quench reactive oxygen species. And then I'll show that this is true also in vivo in a mouse model. So here we go. And this is the induction of SFIA gene that is activated by the generation of reactive oxygen species. And you can see there is much less in the wild type, but in the RPOS mutant missing sigma S, in the presence of GM, the induction is much higher. That's the case with the production of hydroxyl radical, as you can see here. The same thing with hydrogen peroxide, and the same thing with carbonylation of protein. You can see the band is much deeper, and this is graphed here in this bar. And if you then, tested the effect of antioxidants or anaerobiosis, which anaerobiosis does not permit generation of reactive oxygen species, we found that the effect of the loss of sigma S was greatly minimized. And you just need to look at these two slides in here, these two graphs in here. This is wild type plus GM under anoxic conditions. As you can see, it is high, the killing is very little. And it is also the case with the sigma S missing mutant. It's almost as resistant as the wild type, whereas in the aerobic conditions, the sigma S mutant is completely dead. The wild type is also affected, but much less. And then this shows you the individual antioxidant proteins. SOD A, SOD B are quenchers of reactive oxygen species. Same is the case with CATI, and this is a combination of CATI, SOD A. And once again, you can see that the mutant is greatly affected. The wild type is affected, but very little. But the mutant, this is without the antibiotic, this is with antibiotic. In each case, the mutant missing the individual antioxidant protein becomes much more sensitive to the action of gentamicin. Now, these are the quenchers for reactive oxygen species, but the quenchers require a source of electron, and that source of electrons is NADPH, which is generated by the pentose phosphate pathway, and the two enzymes of which are shown in here. And once again, the mutants missing any one of these pentose phosphate pathway enzymes become more sensitive to gentamicin. And here is the situation in vivo. What we did here was to inject UPEC, the uropathogenic Escherichia coli, transuterally in a female mouse, in female mice, there were several. And we found that GM, the administration of gentamicin, prevented colonization of the bladder by the mutant, but not by the wild type. And that the capability of the mutant to, to regain the potential of colonizing the bladder was restored if you simultaneously administered an antioxidant agent like N-acetylcysteine. And these are the the graphs that are showing the same thing. So the conclusions. When bacteria, and even when we respire, when bacteria respire, there is leakage of reactive oxygen species from the respiratory chain. And also, there are many metabolic products that can redox cycle. They are subject to single electron reduction, generating a highly reactive radical that gives this electron to the oxygen and goes back to its original state. And that generates a lot of reactive oxygen species. And therefore, antioxidant defense is required at all times by all of us, including bacteria, to keep ROS, reactive oxygen species, at a manageable level. Now, what GM's done, GM's mitigation, its capability to mitigate the antioxidant defense increases reactive oxygen species to a lethal level, the reactive oxygen species that are generated by these normal processes. And then, in addition, we found that GM itself increases leakage of reactive oxygen species, which is shown by this slide. This is the wild type, this is the wild type, and this is the mutant. Okay, so what do we do about, so this is so you see the GM is a triple whammy. We know that by ages-old studies that it inhibits protein synthesis in bacteria. So that mechanism, exists in normal bacteria, but is greatly impaired in the resistant bacteria. But then GM also mitigates oxidative defense, as I just demonstrated to you, and also it increases oxidative stress, as also I showed. Therefore, damaging the antioxidant bacterial defense will increase GM effectiveness, even in bacteria resistant to 1, which is the normal mechanism of their action on bacteria. And this is the case with the resistant strains of UPEC or many other bacteria. So the question is, how do we go about, mitigating the GM effect on antioxidant defense, the damage to the antioxidant defense of bacteria? I mean, you can't really go about and say that we are going to knock out the sigma S gene in bacteria that are infecting human beings or whatever else. So the answer to that is to find small molecules. And for that, we did a bioinformatics study of the zinc database, virtual screening, and all these programs that technical stuff, but they're commonly used and commonly known. And virtual screening of over 1 million compounds resulted, showed us that we did it for 2 antioxidant mechanisms of proteins. SOD-A is one, and then sigma S itself and SOD-A. And we found that one compound, this one here, was quite effective in enhancing the effectiveness of gentamicin in resistant bacteria. And this shows you this is sigma S, and this is the place where the small molecule that we discovered binds, and this is SOD-A where the sigma S binds, and the antioxidant binds. So, okay, so, great that we have a way of finding an additional way of enhancing gentamicin effectiveness on Earth. But will this be the case in space? You know, we have been working with NASA and they're very interested in this question. Another thing is that, you know, in space, there are all kinds of stresses. Microgravity itself is a stress. Life on Earth evolved in the presence of gravity. And so when you take that away, it's a stress. And one thing that, you know, many things are there, but I'll concentrate on this alone, which is the compromised immunity of astronauts and space travelers. And so, you know, astronauts experience many discomforts and illnesses. And among the most common ones is cystitis, which is caused by uropathogenic Escherichia coli. And when NASA heard that we have been working with UPEC, or uropathogenic Escherichia coli, that causes cystitis, and we have a means of controlling it, they approached us, and we then proceeded to look into it. So the first thing we did was to use a simulated microgravity system, and that you can obtain using these high aspect ratio vessels, or halves. And it depends on the orientation in which they are rotated. This is 1G, normal G control, and this is microgravity. And we found that simulated microgravity causes UBEC to become more resistant to gentamicin, and that this does not happen if sigma S is missing, and that the lack of sigma S continues to make the bacteria more sensitive to gentamicin. But, you know, simulated microgravity is one thing, the actual one is another, and in fact, differences have been found between what people discovered using simulated microgravity and what was found under real space flight conditions. So the acid test was to see whether this thing happens during space flight itself. So in order to do these kinds of experiments in space, you need a color reporter that can indicate changes in the metabolic state of the bacteria, so that the color change can be converted into electric pulses that can then be transmitted from space via telemetry to Earth. And we discovered one such agent, Alamar blue. And here you can see the wild type, you know, the loss of viability as counted by plate counts. And you can see that wild type is really quite resistant to gentamicin, as I have pointed out. The mutant not treated with wild type, as you can see, is as good as the not treated with gentamicin is as good as the wild type, but when treated with gentamicin, it is much more, you know, it is killed to a large extent. And all of this is very faithfully reflected by the change in the color of Alamar blue from blue to magenta. And you can see that wild type plus GM, you know, which is less sensitive, it does become a little bit bluish compared to untreated control. But if you compare it with the mutant, which is very much more sensitive, the blue color is much more intense, as you can see. So it's a very nice system. Okay, so now this is the high-tech stuff that NASA has developed. And it really is is quite a marvel. And what it does is a completely autonomous microfluidic hardware. And, it has the places where you can measure selectivity, which consists of fluidic cards with appropriate number of wells fitted with filters. optical quality membranes, thermal spreaders and sensors for temperature control, 3-color LEDs and photoreactors for conversion of transmitted light to absorbance. And then there is very sophisticated fluid delivery system, which makes it possible for us to control when the nutrient or medium is introduced into the cards, into the wells of the card, when the antibiotic is introduced, and when Alamar Blue is introduced to measure, to find out the changes that have occurred as a result of gentamicin treatment. And that is schematically shown in here. Okay, so this is, by the way, the whole shebang, the complex system harboring this entire system. And what we had done was to put bacteria, UPEG, in a static condition in these cards, and then put it in, the cards were put into this containment vessel, which then was flown to the International Space Station, from where it was launched on November 20, 2017, November 12, 2017. And you can see this being launched. And it's an autonomous system, and from pulses from the Earth, the experiment was started 3 or 4 days after the system was launched to stabilize it. And the medium was pumped to allow the bacteria to grow. Gentamicin treatment was done at 3 concentrations, and then Alamar Blue was added. And as I told you, the change in the color of Alamar Blue was then converted into electrical impulses, which then were transmitted to Earth and were monitored at Santa Clara University. And these are the results. And this is the summary of it. Wild type ground, you can see this is the loss of viability, and you can see it is quite resistant. And this is wild type in flight. What we found is that its resistance to gentamicin went down under flight conditions, which was somewhat different from others had found. But the space reaction, the space experiments are complicated, and a great deal depends on what apparatus, etc., has been used. But the point is that in both situations, the mutant, in the case of wild type, it's in flight, okay. So this is mutant in the ground and mutant in flight. And the point is that mutant in flight retained HGM sensitivity. So elimination of sigma S would be as effective in a space flight as it is on Earth. So this was the kind of thing that NASA was hoping. And that means that the small molecule compounds that we have discovered would be effective also in space. Now a little bit, final few words about space colonization. It's a long-term dream, but as I mentioned, we are polluting the Earth very rapidly. One of the talks by Dr. Hanji, I don't know if I am pronouncing his name correctly, showed how rapidly we are increasing the production of waste. And sooner or later, we may have to leave this Earth behind and hope that we have enough habitable space habitable situation in space. So a lot of work is underway to have autonomous systems that can remove CO2, generate oxygen, grow plants and things of this kind. This is not my specialty, but other people are working on it. And if you like, you can consult this further. So that's it. Thank you.